Good morning, ladies and gentlemen. It's my pleasure to be here and thank you for the invitation. My name is Uwe Marx. I'm the founder and CSO of TissUse, a German-based company which was spinned off in 2010 from the Technische Universität Berlin, my academic background, and since the 10 years is operational in the human-on-a-chip field. Today, I would like to give you an idea how we are going from scientific models based on microphysiological systems toward industrial adoption, and how we try to get into regulatory acceptance of such assays. Our definition for microphysiological systems is microfluidic cell culture devices capable of emulating human biology in vitro at the smallest biologically acceptable scale. We do have much more precise definitions in our community. So I refer you to the next speaker, Susan Fitzpatrick from the US FDA. She will give you a detailed definition for microphysiological systems. And I refer you to the recent MPS stakeholder report, which has been published open access in Altex 2020 last year. In that report, you can find the discrimination between what we are calling an MPS-based model, test, or method on an academic level, what differs for a qualified assay in a commercial setting, and what is finally a validated MPS-based assay. Now, we had the privilege in Berlin here, due to our pioneering role in the field, to host two MPS stakeholder workshops. The second one was in June 2019, and the report which I mentioned for you is available open access, as I mentioned. We were able to combine into that stakeholder report the basic players in academia, like the Wyss Institute, the MIT, Johns Hopkins, and others. We were able to combine here and to invite to Berlin MPS, key MPS suppliers, industrial users, so the end user group, and finally we were able to get regulatory bodies from US, China, Europe, Japan, and Russia to that workshop. Now, in this workshop, among others, clearly highlighted that to get an industrial adoption of MPS-based assays and finally regulatory acceptance, you need a functional value chain. And the value chain for MPS-based assays starts in academia. Academia is using MPS for satisfying curiosity, for novel discoveries, but finally you need a supplier background to produce and qualify the tools and the models. And you need a direct contact into the end user industry, like pharma and CROs, which are qualifying the assays prior to use the assays for patients' benefit by producing new candidate drugs or making precision medicine. And all these should be accepted by authorities, which at the end of the day leads to validated assays, which are regulatory acceptable prior to use for patient decision-making. And we identified in the workshop a number of hurdles between those, within that value chain. The first hurdle is a replication crisis in academia. So academic research often is not reproducible in other labs, but it is still good enough to get ideas and new discoveries along the scene. Now, in the next step, reproducibility and qualification is the most important part. And that qualification starts usually on supplier level, at supplier industry level, with model qualification, biological models, not yet exposure of that models to some substances, but just a biological model. Is it really a qualified, reproducible, long-term stable liver or brain or multi-organ chip? And then these models are exposed to assay development, qualification of assays, where you use that models for repeated dose usage. And here is a big gap between the very young supplier industry, it's not a long time yet, and the very robust qualification tools in pharma and end user industries. And that gap should be closed by communication between both of them. And finally, we still have a hurdle between the data which are produced with MPS-based assays in pharma industry for pipeline decision making and the data which reach regulators, because many of these data are still preclinical data which are used to stop a product prior to getting into clinical trials. So they don't reach IMPD or IND level. And therefore, these data and this information don't reach authorization bodies, regulatory bodies. So that is a kind of the value chain roadblocks we identified, and I invite you to read the report, because we have made a number of decisions and a number of proposals how to get over that problems. Now, an important finding for our community was that the academic development of microphysiological systems is now more than 15 years, or nearly 20 years old, and has a full-fledged big size. So in 2020, we have more than 1,000 scientific papers. And these are all based on scientific models, biological models, methods, and tests which the academia are using. Many of the academia have spinned off later, for the last 10 years, companies. We have a lot of companies now. This is even an old picture. I think we are with 60 companies in the supplier industry now. And these started feasibility testing for the last, let's say, 5 to 7 years with early adapter industries, pharma industries, for safety and investigational purposes. Then finally, you need to get these tools into regulatory science. And we identified that we have at least more than 4 standardized commercial platforms available, which have been transferred to different laboratories, which have been transferred to early adopters, and they are qualified equipment and models here on that level. In pharma industry, the report, our work group, could identify 23 qualified context of use assays, which are already used in internal decision making. Now, internal decision making about a candidate is a sensitive topic and can be done only with a fully qualified context of use. That level of qualification is the highest you can reach. But unfortunately, we could not identify, end of 2019, any MPS-based data submission in IND or IMPD files, and obviously and consequently not even any validated context of use assay which made it to an ICH or OECD guideline. So a lot to do still and a lot of hurdles here, but this is already a success. Here are at least the 4 commercially available MPS platforms. And the beautiful thing with these developments in our community is that all of these companies, from high throughput single tissue organ models to more complex biological models, they all are derived from basic institutes and high level research institutes, and the players which were involved in the University of Leiden, the WIS Institute, the MIT, and the Technical University like myself, are still involved in moving along with that new products and assays. That makes it robust and that makes it scientifically valid. Now, I would like to give you a little information about our alliance of our mother institute, the Technical University of Berlin, and TissUse, where we spinned off TissUse in 2010. So we are 10 years now around, and you see that we were producing on the academic level a lot of master theses, PhDs, postdocs, and 45 peer-reviewed papers along the 10 years. Whilst on the level of the company, we developed 16 human organ models, 12 multi-organ combination out of the 16, and we were able to establish 22 assays. But these are not the key performance indicators. A key performance indicator is always driven by the question, how much of these things are really benefiting the patient? And for the academic landscape, our key performing indicator is a question, how many of these peer-reviewed papers are associated with patients' benefit? Do they really go into a direction of an assay which can be used for patients in pharma industry or in precision medicines? And we had 13 papers which have this link to the patient, which is a very good result for 10 years. Now, even more on the company's level, the most important thing is how many of these 22 assays can make it toward a quality and a robustness and reproducibility, which can be used for decision-making and end-user levels. And in our case, these are six assays which made it to that level. Again, a very good result. Let me introduce our technology for a few minutes to you. So in 2007, we developed our working hypothesis to establish the HuMimic technology. And this is based on two hypotheses. First, each and every human organ is built up by a multiple identical functionally self-reliant structural units. That time we called it organoids, but now artificial organoids are in a little bit different context, so we call it structural units. These functional units are evolutionarily conserved and subject to genetically encoded self-assembly. Such a unit is in the intestine is, for example, a villus. 7 million small intestinal villi are absorbing your food every day. 1 million liver lobuli are processing your food, metabolizing your food, and producing a lot of proteins for your body. And finally, 2 million kidney nephrons are excreting your food. And all of them are of a very small dimension, like millimeter scale. That is evolution, and that is important to know for our strategy. Now, in 2012, we published our strategy, how to build entire human organisms on chips with a downscale factor of 1 to 100,000. So 100,000 for smaller human on a chip. The reference organ is liver. We decided to go for a capacity of 10 liver lobuli in the small liver compartment of such a chip. And we found out that we need to combine another at least 9 different organs in a right ratio. 10 liver lobuli need 70 small intestine villi. They need 3,000 lung alveoli combined in a microphysiological kind of blood flow in order to compose a full minimal organismal biology. Now, whilst we are still working on that HuMimic XX or HuMimic XY full-blown organisms, male and female organisms, we used the same design, the same chip design to make easier to use chips, like chips for 1 or 2 organs, chip for 3 organs, and chip to interconnecting 4 organs. These chips and these equipment, qualified equipment to IQ, OQ, PQ standards in Europe, these are going into academic labs, and academic labs are using them for their own modeling. So no biology associated. But more interesting, we then started to implement qualified biological models into their chips under GCCP, good cell culture praxis, good cell culture praxis standards, which have been published in 2018. Now, the models are qualified for at least 28 days, and this is not yet an assay. Finally, to make it a qualified assay in context of use, you need to develop an exposure regimen, the repeated dosing schedules, and other things. So that is the third level of qualification in our company. And finally, regulators' approval or registration should go on the basis of a qualified context of use assay. To make that a high quality, we qualified our company under a total quality management according to ISO 9001:2015. Now here, in a nutshell, is the unique selling point of the systems. We have implemented a micro pump on the chip, so it's like a small heart, and that produces a pulsatile physiological fluid flow through one or two, in this case, two cell culture compartments. On this particular chip, you have two circulations, so you can make two repeats. These are size of a standard microscope slides. You can use primary and iPS-derived cells, 3D constructs, and biopsies. And finally, this all is compatible to live tissue imaging from below. And you have an option to plug in insert-based barrier models into that system. And that is a transfer to any laboratory in the world which want to use that technologies. Now, our assay portfolio, as I told you, includes 22 different assays as of today, and it is growing stepwise. We have defined, according to the qualification level, we have defined 3 levels of readiness within that assay portfolio here for the different context of use assays. Now, the first level of readiness is academic biological model, very early developed model with a few specifications only, useful for scientific discoveries, but not yet qualified at a scale which is acceptable for commercial use or for decision making for patients. Now, the next level, again, is then model qualification level 2, that are qualified humic models, biological models like these ones shown here, which have already a standard in-process handling and a number of clear specifications for reproducibility. The biological model is established, and once it is established, the last level is qualify an assay using that particular model. For example, having intestine, liver fully as a model, but now developing a repeated dose 7-day exposure through the intestinal barrier into that model. That is the last level of qualification, and that then allows the end users to make candidate decisions internally after technology transfer of such assays to their laboratories. Now back to what our findings within the stakeholder community. These are the 23 cases we identified which have made it to decision-making worldwide in a global context. And we are proud that 6 of these assays are coming from our laboratories. And I just shortly want to introduce to you, for example, the bone marrow on a chip assay development as a case study for how these things can evolve. Now, the functional bone marrow unit is a spheroid volume in a spongious bone space where a capillary goes in and out. You have an oxygen gradient, and here you have mesenchymal stem cells and hematopoietic stem cells, which are forming a hematopoietic stem cell niche, and continuously cells, white blood cells, are developing from that and red blood cells and getting into the bloodstream. Here are the different cell types which are produced by that hematopoietic stem cell. We were able to model that hematopoietic stem cell niche in our chip-based humanic platform by a pulsatile flow with the same using mesenchymal stem cells and hematopoietic stem cells in that niche. This was an academic model first. We published it in 2018, and these human hematopoietic stem cell niches were surprisingly stable. Over 4 weeks, they continuously produced all colony-forming unit assays. They were able, when you transplant after week, these cells from the chips into knock mice, you could show after weeks that human white blood cells are coming out of this mock cells. That intrigued AstraZeneca, the Drug Safety and Metabolism Department, to jointly develop a specific context of use assay, which takes us 1.5 to 2 years. And that context is repeated dose hematopoietic lineage toxicity testing, scheduling of hematopoietic treatment schedules. For that particular qualification, we needed a number of steps to go through. We prolongated the culture period from 4 weeks to 8 weeks. We secured commercial, robust, and reproducible supply of cells. We optimized and reduced the costs during running the model. We made an in-depth model characterization of resident cells in the bone marrow and those cells which are released into circulation. And finally, we performed an interlaboratory qualification with our partners in AstraZeneca prior to tech transfer. The assay was so cool and so well established that immediately other pharma companies started to jump on the same assay. It's important to mention here that the assays which we are developing together with partners, no matter whether it is industry or academics, are then open for everybody because we want to translate them into any laboratory in the world and to make it available for everybody. Now, let me, for the last minutes, give you an idea about what our community is thinking about the roadmap into the future. We have developed a 15-year roadmap as of today. So we think that within the next 5 years, both the end user and the regulatory scene will grow up to a full-fledged scale, like the other two too. We hope that we can really transfer models from qualified assays in end-user industry into IND and IMP data sets. And that will feedback also into the end users from authorities because it is an exchange here. Now, I showed you that we are really running an back and forth communication between the suppliers and the end users and between the academia and the suppliers. So a lot of things will happen in the next time. But the important thing is, another important thing, let's say, is what are the drivers? And we think that the largest drivers for that kind of technologies are advanced therapies such as CAR-T. If you are using human cells to treat human patients, you cannot go for animal models longer because the species differences are really relevant there. And the MPS scene here can benefit from that advanced therapies. And the last question was, how long does it take us to come up with organismal solutions, which I introduced to you early in my talk. And again, we also see here at least a 10-year time frame to get into more organismal homeostasis. I invite you to read the ideas in that papers here. And then finally ending up with MPS-based clinical trials in preclinical settings. But it will take us another long time to bring that assays into ICH guidelines at the regulatory level. So with that, I would like to conclude my presentation. I hope I could make clear that at the academic level, MPS-based scientific models, methods, and tests support new discoveries and add to the investigation of mode of action of drug candidates and therapies. But it needs a qualified context of use assay and a higher level of qualification to get these assays into safety and efficacy testing with clear candidate decision makings and decisions which are related to patients' benefits. And finally, validation of such assays should be a joint effort of all stakeholders in the next years to get this into place. And here I refer you to the next presentation of Susan Fitzpatrick from the US FDA. If you want to know more information about those assays which made it to decision making, especially those ones which are multi-organ assays, liver pancreas combination, liver thyroid combination, skin tumor combination, please always contact Dr. Reich Holland, our CEO, under that background. And if you want to know anything more about the other 22 assays, keep going with that contact. I would like to thank you for your attention, and I would like to thank a number of academic supporters like the German Ministry of Education and Research, the Eutox Risk Project, the HIPSTAR. On the other hand, our industrial partners, AstraZeneca, Bayer, Roche, all which are like open to show and are early adopters and are open to show this data to the public domain. And finally, I would like to thank the partners which are fostering approaches for regulatory acceptance of MPS-based assays globally in the FDA and in China, because we think that harmonization of that activities are the next big step into benefit for patients. With that, I thank you for your attention.